Hi, in this video I am going to explain some model questions for competitive exams to the pharmacy students. If you like this video, please subscribe to my channel for more videos. Question 1. Which complex of an electron transport chain is inhibited by carbon monoxide? So you can observe the complex 1, complex 2, complex 3 and complex 4 and complex 5 in the electron transport chain, ETC chain we can observe. So is the one broad view of complexes we can observe in the ETC chain. So in the complex one we can observe the FMN is converting into FMNH2 by taking off 2 electrons from the NADH and it is converting into NAD. So here the iron is going to help. And in the complex 2 we can observe the FAD is converting into FADH2. So here the succinate is converting into fumarate. Succinate reductase enzyme is involved here. So this FMNH2 and FADH2, what are the two are formed in the complex 1 and complex 2. So the electrons are transferred to the complex 3 through coenzyme Q. So this complex 3 having the cytochrome B with iron. So here the electrons are transferred through the iron to the cytochrome C, and this cytochrome C transfers the electrons to the cytochrome complex 4 having cytochrome A and cytochrome A3. So from this the electrons are transferred to the oxygen, and by taking off H plus ions, this oxygen is converting into water molecule in the complex 4. And in complex 5 we can observe the ADP by combining with the phosphate, it is converting into ATP by the ATP synthase. So here in this first process we can observe the NADH CoQ reductase is involved here, and here the succinate Q reductase is involved here. And in the complex 3 you can observe the cytochrome reductase enzyme is involved here, and complex 4 cytochrome oxidase is involving here. So among all these, the carbon monoxide inhibiting the cytochrome oxidase. So all these three are reductase, so it is not going to influence the carbon dioxide, or the oxidase enzyme only influenced by the carbon monoxide. So the answer is the complex of electron transport chain is inhibited by the carbon monoxide. That is complex 4 cytochrome oxidase. Question number 2. Clarithromycin is recommended for the treatment of. So given the options are filariasis, leprosy, malaria and whooping cough. So here we can observe the filariasis. So mostly the recommended 3 drug regimen is recommended for the treatment. So ivermectin, diethyl carbamazepine and albendazole. In the case of leprosy, so the combination of rifampicin, clofazimine and dapsone is mostly preferred. In the case of malaria, WHO recommends the parenteral artesunate in preference to quinine for the treatment of severe Plasmodium falciparum malaria in adults and also in children. And in the case of whooping cough, we can observe the azithromycin, clarithromycin, erythromycin and sulfamethoxazole are mostly preferred. So clarithromycin is recommended for the treatment of whooping cough. Question number 3. Which of the following is used as a preservative for blood glucose? So among these 4, if you are observing, the sodium fluoride is going to completely inhibiting the enolase enzyme in the glycolysis pathway. So by this we can see the conversion of 2-phosphoglycerate to phosphoenol pyruvate. So this step is completely prevented by the inhibiting the enolase enzyme. So there will be there is no conversion of glucose we can observe. So acting as a preservative, sodium fluoride. So answer is the sodium fluoride. So which of the following is the correct order of stability of alkenes? So here we can see the different orders are given. So but basic thing, the order of stability of alkenes we should consider is the substituents present in the alkene, the number of substituents present in the alkene. So if it is more substituted, it is more stable. Less substituted, less stable. So you can see the order of stability alkene I represented here. So basing upon this basic formulas of alkene, we can easily find out which one is having the more stability and less stability. So here I given the structures, that is iso, that is this structure of butene, isobutene. So here you can observe this is clearly indicating this type R2C CH2. So this is that one. And next we can observe this trans butene and cis butene. So you can observe the one carbon is having one substitution another carbon is having the another substitution. So this trans and cis are coming under this category. And one ene, so it is coming under this one. R, CH, so here R, one substitution, one hydrogen, double bond, CH2. So order of, you can observe the iso is having the more stability than with the trans and cis. So among trans and cis, we can observe the trans is more stable. We know that trans form is more stable when comparing with the cis form. So iso is greater than trans, trans is greater than cis, cis is greater than 1 ene. So this is the order of stability we can observe. More substitutions, more stability. So here the answer is the iso, trans 2 ene, cis 2 ene, 1 ene. And fifth question. So which reagent used in the chemical test for alkalides yield yellow precipitate? So all these tests mentioned in these options, Wagner's reagent, Dragendorf reagent, Hager's reagent, Mayer's reagent, all are using for the alkalide detection only. So among these, the yellow precipitate is produced by the Hager's reagent. So we can see the composition and what is the result we can observe by using these reagents. So Wagner's reagent for 250 ml, the composition if you are observing, so 2.5 grams of iodine and 12.5 grams of potassium iodide is present and used to detect the alkaloids, and we can observe the reddish brown precipitate is going to appear. And Dragendorf reagent, so for 100 ml preparation, so we are taking the bismuth subnitrate, that is 1.7 grams, and glacial acetic acid 20 ml and water 80 ml and 50% solution of potassium iodide in water. So this mixture we are calling it as Dragendorf reagent and used for the detection of alkaloids, and it is going to produce orange red precipitate. So in case of Hager's reagent, so it is prepared by 1 gram in 100 ml of water of picric acid, saturated solution of picric acid. So used to test the alkaloids, and it is going to give yellow color. That is the previous question what we observed. And Mayer's reagent, so for 100 ml preparation of Mayer's reagent, we should take that 1.36 grams of mercury chloride and potassium iodide 5 grams we should take, and used to detect the alkaloids, and it is going to give the color is the cream colored precipitate. And question 6, what is the approximate molecular shape of phosphorus pentachloride? So we know that phosphorus pentachloride PCl5, it is a trigonal bipyramidal we can observe. So here I am representing the some of the shapes I represented, molecular shapes. So trigonal pyramidal we can observe for the phosphorus trichloride PCl3. So having the bond angle 109.5 degrees as like tetrahedral. And trigonal bipyramidal, that is the phosphorus pentachloride. What is the question now? That is phosphorus pentachloride. It is a trigonal bipyramidal. It is a shape we can observe. And we can observe the linear, that is carbon dioxide. We can give the example having the bond angle 180. And in the case of bent, we can see the example, that is sulfur dioxide. So having the bond angle 119.3 or nearly 120. And ammonia, we can observe the trigonal pyramidal. So this same as like this phosphorus trichloride. So trigonal pyramidal NH3. And we can observe the trigonal planar. Trigonal planar, we can see the BCl3 boron trichloride. And T-shaped also we can observe, that is bromium trifluoride we can observe. So these are the some molecular shapes we can observe. Here our question is the phosphorus pentachloride, it is having the trigonal bipyramidal. Question number 7, iodoform is prepared industrially by. So iodoform, that is CHI3. That is the formula of iodoform. So if you observe all the options, so first one is the chlorination of methane. So it is chlorination, not iodine is adding. So electrolysis of an aqueous solution of ethanol containing sodium carbonate and potassium iodide. So potassium iodide we are adding to the sodium carbonate. It is going to give the iodoform. And third option, that is by heating ethanol or acetone with a bleaching powder or with a chloride and alkali. So it is also not going to produce iodoform. And electrolysis of an aqueous solution of ethanol containing a sodium carbonate and potassium bromide, not potassium iodide, they given the potassium bromide. So the correct answer is the B option. And question 8, the drug mercaptamine is used in the treatment of which condition? So the given options are cystinosis, cerebral malaria, hepatitis, and infections caused by the gram-positive organism. So we can see this is the cystinamine, that is nothing but mercaptamine. So another name for the mercaptamine. This is the structure of mercaptamine. And cystinosis is a rare genetic disorder that causes an accumulation of amino acid cysteine. So that's why the cystinosis, the name came, within cells. and forming crystals that can build up and damage the cells. So these crystals negatively affect the many systems in the body, especially the kidneys and eyes. And what is meant by cerebral malaria? It is a diffuse encephalopathy associated with the seizures and status epilepticus, which can occur in up to 1/3 of patients with severe malaria, particularly that caused by the Plasmodium falciparum. So in the most of the malaria conditions, we can observe the cerebral malaria. We cannot observe with the mercaptamine. And hepatitis, we know that it is a liver toxicity. So liver damage we can observe by the hepatitis B virus, and infections caused by the gram-positive organism. So the mercaptamine, it is causing the drug mercaptamine, it is used in the treatment of the cystinosis. The answer is the cystinosis. And question number 9, what happens when gelatin is treated with the soda lime? So the given options are: it turns the brick red color, there is a no identifiable change, ammonia evolves, it gives a white precipitate. So when the gelatin is treated with the soda lime, you can see the ammonia is going to evolve due to the nitrogenous content is present in the gelatin. So here we can observe the different tests we can observe for the gelatin. So among that, first one that is related to this question. When gelatin is treated with the soda lime in dry test tube, ammonia is evolved due to the presence of the nitrogenous compound in gelatin. And gelatin solution is added to the Millen's reagent, it gives a white precipitate, and by heating it turns to red color. And gelatin gives a buff white precipitate with the tannic acid solution. And in the biuretate test, 3 ml of test solution of gelatin with the sodium hydroxide is added, whereby white to whitish buff colored precipitate is formed, which does not dissolve on heating. And we can observe the yellow precipitate is forming by addition of the picric acid to the gelatin. And also similarly, we can observe the yellow precipitate by reacting with the trinitrophenol in aqueous solution. So these are the different chemical tests we can observe for the gelatin. So by reacting with the soda lime, it is going to evaluating the ammonia due to the presence of the nitrogenous compound in gelatin. 10th question, which macrolide is formed by semisynthetic conversion of erythromycin? So here we can observe the azithromycin and clarithromycin. These two macrolides are derivatives of the older macrolide, that is the erythromycin, and remaining are not forming from the erythromycin. And they are used in the treatment of variety of infections, including community acquired respiratory tract infections and mycobacterial infections, mostly using of this azithromycin and clarithromycin or erythromycin we can observe. So the answer is the azithromycin. If you like this video, please subscribe to my channel and share to your friends. Thank you.